Let's do an entrance exam. Who's brave enough to tell me the difference between an axon and a dendrite? You were first.
An axon's the outbound pathway, dendrite's the inbound pathway.
Exactly. So a neuron is a neuron. To first approximation, don't tell my wife this, who studies connections between dendrites, but axons send outgoing signals that are received by either cell bodies of neurons and dendrites, and those connections are synapses. Okay, you're ready. So I study primarily the cerebral cortex, which, again, a bit of remedial, I'm sure you all know this. The cerebral cortex is the shell of cells and connections on the outside of mammalian brains. In us, in the human brain, they're pushing 100 billion neurons in the cerebral cortex. In the mouse, which is a factor of 10 smaller, and because of the cursive 3D, and you'll see why I say that in a minute, it's 1,000 times smaller. So every time you go down a factor of 10, you have 1,000 times more data you might collect. But we, the first approximation, think, our thoughts, our percepts, our planning, our most of what we think we do with our brain is with the cerebral cortex. And remarkably, the fundamental building blocks of the cortex and the architecture are very similar between man and mouse and any other mammalian species, not just the cells and cell types, but the layers and the statistics of connections to first approximation, which is why many of us, perhaps most of us, study the mouse, and why at the Allen Institute for Brain Science we study actually both the mouse and the human, trying to find connections between the two. And the next question and answer is opinion, but I think it's pretty well-founded opinion, that what determines what the cortex does, or really any brain circuit does, it's the connections. There's a huge amount that goes on with the dynamics and coding strategies that falls under the category of physiology. There's anatomy and physiology, but it's really the connections that makes the brain compute, that make the brain compute. And another opinion, but it's an opinion that really I've spent my entire career on, and many of us have, that we'll understand the cortex or any brain structure when we have a wiring diagram in a functional context. You need one, but you need both. And whenever I give a lecture, the thing that people remember the most when I show it is a movie from now 50 years ago of the way that people back then and still today predominantly study activity of the brain in a living brain. And this is an experiment. I don't have their names here, shocking. Who has heard of Hubel and Wiesel? It's almost one word now. They were at Harvard in the 60s, and they really laid the foundations for the study of the visual cortex, but really laid the foundations for many, if not most, people's thinking about what simple computations in the cortex might look like. And what they did was they studied the visual cortex, the visual part of the brain, which gets input from vision via our retinas and our eyes, via a relay to first approximation in the thalamus, and then sending relatively unchanged information from the eye onto the visual cortex. And it's an extraordinary thing to be encountered with the activity of a single cell in the brain, particularly in a sensory system or in a motor system, where you can really get a sense of input and output. So what you'll be listening to are clicks. It sounds like Geiger counter clicks or something. And those are individual action potentials, the individual firings of one neuron. And that neuron is quite literally seeing. And what is it seeing? It's seeing a piece of paper on an easel in the 60s. And you'll pretty quickly get a sense of what it's seeing. The sound was there. Oh, good. So you can hear what it does. Click, 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 click. Torsten and David are drawing. Oh, tragedy. Let's see if this works better. I transcoded this last night from a format, not quite from the 60s, but an old format. Is this any better at all? Should I do it? It's fun. Cubal, Wiesel, direction, and if this doesn't work, let's see if this works. It's louder. But you can pretty much see it. Whenever the bar is in the right place, the cell fires. And I guess we're not going to see it, but I'll pretend I'm the little bar. And what they discover is when the bar moves from right to left, the cell fires. So it's a right to left detector. And other cells in the same part of the brain will respond to motion, or not to motion, but to vertical bars, horizontal bars, light, dark. In us, there are cells that respond to color, etc. So you can think of this as a computation, and then the question is, how is the thing wired up in order to do this computation? And then you can imagine there are two questions. Again, I've already said it. What do the cells do? What do the cells do? They see something, for instance, and how do they do it? How are neurons organized in the brain? But really more importantly, what's the wiring diagram? Enough of this. An analogy, and maybe a nice analogy for where we are. Nowadays, we study the brain with electrophysiology, but also with imaging techniques that you'll see very, very soon. And I like using a computer analogy. And using an example from the origins of computer science, the Colossus computer that was used in Bletchley Park to decode the Enigma codes. Colossus reverse engineered the Enigma machine, the encoding machine that the Germans used. How might one reverse engineer Colossus? And you could look at the instrument panel and get a sense that it's doing some sort of counting. But there's Alan Turing, just because he was the mind behind much of this work. If you're going to understand how the thing works, wouldn't it be great to have a wiring diagram? And I found this somewhere. This is the wiring diagram of of a decimal, a biquinary counter circuit. So clearly it's doing some kind of decimal arithmetic that you can get from the wiring diagram. Nowadays we have almost the same thing. We have the ability not to listen to one neuron at a time, but the ability to see hundreds, if not thousands, and really up to tens of thousands of neurons in a brain circuit. And this is the flashing of neurons corresponding to action potentials in them, collected with two-photon microscopy, a kind of nonlinear laser scanning, nonlinear optics, laser scanning microscopy. And the label here is calcium, which is a readout of neural activity. Wouldn't it be great to see what thousands of neurons do in a cortical circuit, and then to look into the cortical circuit and for exactly the same neurons try to figure out what are the connections and what's the logic of the connections with respect to the computation that's going on. Please ask questions if I'm being obtuse, or if you have questions. You'll see more of this later on of the kind of data one can collect and analyze nowadays. But those are brain cells. Connectomics. It's not everyone's favorite word, but it's the word that we're stuck with in the field. It's sort of back formation from genomics, where omics has become in biology the science of complete data sets and complete overviews of any given field of study. Connections, omics, Latin, Greek, sort of bastard Greek at that. It is what it is. But, the field really started about 15 years ago. There had been, modern neuroscience began with anatomy in the 1880s, 1890s, and beyond. There has been a great deal of neuroanatomy done with light microscopy, and in the 60s and 70s, electron microscopy really revolutionized our understanding of neurons, their structures, their subcellular organization, and to a certain extent, their connections. Really from the beginning, there had been three-dimensional electron microscopy. And how do you get 3 dimensions with anything? There are 2 ways of doing it. One is to scan into a 3-dimensional object and only be able to see certain objects within it. The other is tomography, slicing, taking 2-dimensional views and adding them up in 3 dimensions. So serial section electron microscopy, starting in the 50s, late 50s, was what biologists and largely neurobiologists used to study the 3-dimensional structure, all the way down to the finest details of nervous tissue.
How reproducible is that between human beings? In other words,
Yeah.
it's the individuality, obviously, but at the same time, is there
Yeah, I mean, this is sort of the first question, and it's a great question. When you're in biology, how much is reproducible? What are the idiosyncratic details of a single organism, a single cell, versus the general rules? And like anything else, you have to use your judgment, and eventually as the field matures and you can do comparisons, you can address that question head on. I think in any fields, I won't be general, in neurobiology, you have to use your wits to figure out what to do. Every single detail, of course, is idiosyncratic. It differs between individuals, it differs between neighboring cells when you're studying a circuit with 100,000 cells. So look for the rules. Look for the strict or not strict rules in neighboring cells, between individuals, and things that reasonable arguments would say these are general principles versus they're not general principles. So yeah, if we are studying, as you'll see, a network with 100,000 cells and 1 billion connections, how could you possibly do one of those? But by the same token, how can you possibly do one genome when you know that everyone has a different genome? You use your judgment and use the literature and use just simple arguments. If I get to it by the end, I can give a simple kind of argument to regularities that, if found, give a rule and should be found everywhere. Question?
I feel like that might be better on different.
Okay, I'll take that. But the modern field started 15 years ago with Winfried Denk, then in Heidelberg, who automated serial section electron microscopy. I won't tell the details unless people ask later on. And if you've read the 2 articles I signed, the first article discussed the different techniques to do it. But let's just say magic, very difficult, laboratory, very difficult experiments. But what you get at the end is a series of 2-dimensional images that look like they're evolving in time, you'll see soon, but are really just marching through in the 3rd dimension, Z. And right here you have a nucleus. How many people have looked at electron micrographs of the brain? Okay. Maybe 1, 2. That's a nucleus. It's a big round thing. Here's another nucleus. Here's another nucleus. You might be able to see a little line there. That's a dendrite. As soon as I animate it, how am I doing with movies? A+. As soon as I animate it, you'll be able to see, if nothing else, a little bit of motion. Let's watch this thing evolve up. Did you see that? Do you see that thing coursing? Do you see that thing? So you see motion. What those are the larger neuronal wires, big dendrites, going in 3 space, showing their 3-dimensional structure as you march through the 3rd dimension. And what this showed was that, it challenged us. Why not bring 3-dimensional electron microscopy into the 21st century, where we have computers and we have extremely fast data collection devices. Next are a bunch of slides I show in audiences like this that give a sense of A, neural structure, and B, the sizes of data that we need if we want to study structure that do 2 things, create data sets that do 2 things. One, show the finest details. If we want to study synapses, the connection between neurons, we better see synapses. If we want to study wires, axons in particular, we better have the resolution to see individual axons. Individual axons get as small as 50 nanometers across, well beyond the resolution of light. Really only electron microscopy can do this. And if we want to study individual neurons, then neurons are 100 microns around, circuits are order a millimeter, brains are big. If we want to study small things and over multiple orders of magnitude, you need to collect a lot of data. This is the first high-resolution electron micrograph you'll see. It's roughly a micron by a micron, or a micron and a half by a micron. You might see a bunch of circles. Those circles are vesicles with neurotransmitters in them. So the vesicles are tiny, 40-ish nanometer sacs of chemicals with a membrane around them. They're in the axon, that sends signals. When this neuron is active, its cell body over 100 yards away at this scale, its cell body fires. It sends a signal down the axon. The axon does things that make these chemicals go in this thin synaptic cleft, and then there are proteins in the dendrite that receive that signal. It's like a transistor, essentially, that this is the gate. It sends the gating signal, and that allows current to flow, actually, from the outside into the inside of the cell to depolarize the cell, to activate the cell. Or it might inhibit the cell for different types of neurons. At 1 micron, a cubic micron in the brain, has 1 synapse on average, okay? And the resolution one needs to study these things are order 10 nanometers. So this is going to be 7 slides in a row, powers of 10. If we're doing 10 nanometers into a micron, that's 100 by 100 by 100, a million data points. Okay, so even to study a synapse in the old days, that would be a lot of data, a megabyte of data, and you'd study 1 synapse. But that doesn't tell you, that's not connectomics, that's just a little bit of solder in your wiring diagram. Let's go up by a factor of 10. But again, a factor of 10 is a factor of 1,000 in volume. Then we see roughly a cell body. 10 microns is 1,000 times 10 nanometers. That gives you a gigabyte of data at that resolution. This isn't much of a circuit. This shows all the synapses onto the cell body and a few of the synapses from the axon that belongs to this cell body. In a cubic 10 microns, there's on average 1/10 of a neuron in the cortex and 1,000 synapses. Okay? Still not a circuit. Let's go up to the scale of individual neurons, or at least the proximal, the core of a neuron. And this is 100 nanometers again. In a 100 nanometer box in the brain, you get 100 neurons, 1 million synapses, a ton of data, a terabyte of data. And you might be able to study you might think that you could study well the connections between these 100 neurons. But most of the output connections from this neuron live elsewhere. Most of the input connections to this neuron come from axons elsewhere. So really you can only begin to get into this game of 3-dimensional reconstruction with a terabyte of data. But that's the beginning, and that's where we were, or in between, maybe a couple hundred of these 8 years ago. We were able to look at a couple hundred of these. So, big idea, which I've certainly beaten over the head, is that to study the small over large scales, you need a lot of data. You need the large scales if you're going to study circuits. This is really the goal that we've arrived at now. But at the beginning of this field 15 years ago, this was the grand goal. Are we ever going to get to a cubic millimeter? Are we ever going to get to an entire local cortical circuit with 100,000 neurons, 1 billion synapses, and largely self-contained, except for the long-distance connections to and from this, but largely self-contained. It's a petabyte of data, but as of the end of last year, we've collected a data set like this, and our colleagues at Princeton, Sebastian Sung's group at Princeton, are helping us analyze it. They used machine learning to analyze the data set and take the impossible to see, let alone use, 3-dimensional black and white data and segment it into axons, dendrites, synapses, and ultimately a graph.
Once you segment it, the amount of data is smaller.
The amount of data is, There are not many empty spaces. Well, there are. Let's ignore the empty spaces. The data, a petabyte is big, but it's manageable now. By segmenting it, you do reduce the data. Ultimately, if all you care about is connectomics narrowly construed, all you want is a 100,000 by 100,000 matrix with mostly zeros and weights of connections, input-output matrix. Alternatively, you might want a three-dimensional structure of every neuron. Still, that's highly reduced. You might want every detail, and then it's reduced, but not radically reduced. A mouse brain, that's what we're. It would have been enough at a cubic millimeter, but people today, and I'll get to this at the end, are thinking about doing a whole mouse brain as a grand challenge, because why not? More than why not. It's really, can you imagine having the whole thing? I'll talk about it later, but you can see why it's exciting. It's a whole brain. Every single connection between every single neuron, the morphologies, the morphological cell types, and it's roughly 100 million neurons, roughly 10 to the 12 synapses. These numbers are wrong, so neurobiologists listening in the sky, none of them are wrong by more than a factor of 2, and today we're physicists. So, but it's a lot of data, an exabyte of data. But that's, exascale science exists. Accelerators, collect data at the exascale. Most of that is very quickly reduced in size, but collecting that much data is doable.
When you get something like this, you don't have a functional correlate yet, right?
Wait.
What is the comparison of scale? How big is the internet?
Google in 2018 had 14 exabytes. At commodity prices, that's $20 million of disks. I think that's right. But reduce it. So thank you for that question. If it can't be reduced, we're doing something wrong, and we're not going to do this. Let me keep going. This is kind of silly, but at the scale that I'm talking about, 10 nanometers, the whole human brain, a zettabyte, the internet I don't think is anywhere near, maybe it's a zettabyte, who knows. But no one wants to fill up the internet tomorrow with the human brain's data. But go back by a factor of 10 in resolution, 100 nanometers. Very small, and if I have time, I'll tell you that's an interesting project. That at 100 nanometers, you can really get a lot of the important structure, and that's again only an exabyte. And if that scares you, go down by a factor of 3, and then you're down by a factor of 27 from that, whatever that might be for petabytes. So from that paper, I'm going to speed up now. I'm dawdling. The promise was to image neural tissue in 3 dimensions to reconstruct every axon, dendrite, and synapse. Connectomics, as defined by Sporns in 2005, is a comprehensive structural description of the network of elements and connections forming the paren human brain. Okay, what I said it was. Fixed cells, yeah. This is not dynamic. Absolutely. If I really speed up and skip some things at the end, I deeply believe that if anything in neuroscience will constrain our understanding of learning rules, it will be connectomes. that these things have to have been learned, and they'll have such extraordinary structure that I think it'll be an amazing, provide inroads into understanding what learning rules had to have been there. And we can see some signs of plasticity too, which I'll show.
Related to this question, so when you are imaging it, is the brain doing some task, or is it like quiet?
I'll get to that again. Thanks for asking. So, sorry if this is advertising, but anyway, the first real connectomics papers were in 2011. 7 years after the methods paper, and side by side, we had a paper. Danku did the 2004, had a paper, and there were very, very similar papers. There were first calcium imaging, seeing what neurons do in living tissue, and then reconstructing networks and asking specific questions about the relationship between wiring and firing, neuronal firing. We're on the cusp of very large-scale connectomics, and just from our group, I'll show how quickly things have moved. But there are 4 or 5 big labs in the field, and by now almost a dozen groups doing this science. In 2011, we had a lot of data, but everything was traced by hand, and we were only able to look at a couple hundred connections. But we followed our noses, cared about the neurons that we had imaged, that we had seen what they do in the living brain, to address questions about what those connections might look like in those neurons. And we got some science with only 200 connections. 5 years later, slightly larger data set, a fair amount more physiological imaging, and a lot more student time of tracing things in 3 dimensions, around 1,000 connections amongst 100 plus neurons. And that was sort of the state of the art. There were multiple papers by this time, and everyone was working in the small number of thousands of connections back only 3 years ago. And this is the paper I had you read, and it showed, consistent with other studies, similar studies with different techniques, that neurons that do the same thing and are near neighbors tend to be connected to each other. It sounds like a very obvious things, but in neuroscience, where there are a lot of theories and not so much data to support or disprove theories, there's always, 5 years prior to this, people said absolutely not, that these excitatory connections are random with respect to what the neurons do. Go figure. We and other groups showed sort of the obvious thing, that if there are neurons that do completely different things that are right next to each other, so they could connect to each other, they congregate in sub-networks that do the same thing. They reinforce each other.
What makes a neuron, or what pushes a neuron to connect with one and not with the other? It's a random process that they
Yeah.
looking randomly and then connect like that.
That's a good question, and thank you for asking it. We all know Hebb. No. Donald. So there are lots of learning rules. Oh, here's the question I meant to ask at the beginning. How many people have do machine learning here? So some fraction, and you've already had lectures on machine learning, and you live in 2019, so you know about machine learning. There are, in neuroscience, there are lots of learning rules. The father of all learning rules is Hebb's rules, Hebb's rule. And I'm going to give, again, a wrong definition of that, but it's one that's easy to remember. It's neurons that fire together, that are coactive at the same time, wire together. So there are all sorts of learning rules in the brain, and it's a huge field, but standing back a bit more, there are, let's say, 3 determinants of connectivity. First and foremost is geometry, that if you're not in the right neighborhood, you're not going to connect. Second is molecular, and sometimes people call this, I like to call it cell type connectivity. There are molecular signals that neurons send and receive, many of them on the membranes themselves of the neurons, that make certain neurons or certain membranes like to touch each other and connect to each other. So there can be molecular determinants, then there are activity determinants. And what I just told you about Hebb's rule and all the following things, those are activity-dependent learning rules. So this almost certainly is not molecular, although people still like to argue that it might be, but it isn't. This is almost certainly activity-dependent learning rule.
You said they fire together, they wire together. You're saying if there is some signal that causes neurons to fire and they're maybe close enough to each other, you're saying that they're more likely to connect?
Yeah, that's the right interpretation of my wrong statement. Yeah, the better statement, the classical Hebb's rule is if neurons are next to each other or already have a weak connection, let's say they have a weak connection, that if neuron A that provides the connection, axon, fires right before neuron B, that connection will strengthen. And in this way of thinking, one very standard model is that neurons connect promiscuously, densely at the beginning, and if the connections are weak and are not strengthened, they'll disappear. That's one model. Again, stop listening. All neurobiologists who are listening to this, I'm talking vaguely. We're part of a program funded by IARPA, which is like Defense ARPA, Research Intelligence ARPA is the intelligence version of DARPA. And this program very nicely funded a large-scale effort to do precisely this experiment, actually, to do calcium imaging first, followed by electron microscopy at the millimeter scale. And in 2019, we have, as you'll see, this data set with a cubic millimeter with 1 billion connections and 100,000 neurons. And while we're a good way away from the connectome, we are able, or Sebastian Sung's group at next door at Princeton is able to first approximation, identify these 100,000 neurons, and identify on the order of 1 billion synapses. Wiring it all up, that's work for the next couple of years. Why did they spend this money? The goal was to combine neuroscience and data science to understand cortical computations. Why? To deploy mathematical abstractions of these operations in novel machine learning algorithms. A lofty goal, we'll see if we get there. But it is absolutely true that machine learning, especially convolutional nets, but much of, if not all of machine learning, was inspired by neurobiology, and really inspired by neurobiology, starting with Hubel and Wiesel in the 60s and really ending with people who studied learning rules in the 70s. There has not been much of an infusion of neurobiological ideas into machine learning, the first approximation in the past 20, 30 plus years. Because with enough primitives, you can do an awful lot of things. But if you have an entire circuit, that might allow you to see motifs, et cetera, that you just wouldn't have thought of from learning rules alone. I always run out of time. This is our group at the Allen Institute. Fearless co-leader Nuno da Costa, who is an electron microscopist and has been doing science like this for quite a while and is really leading the experimental effort that includes Joanne Buchanan, Dan Bumbarger, Agnes Bador, Wenjing Yin, and there's an analysis effort, Forrest Coleman and Casey Schneider-Mazel, whose work I'll be showing at the end, and she's our project manager, sort of keeping this large project going. And we're part of a consortium of 3 groups. The first group at Baylor College of Medicine that did the physiology for these experiments and is also doing the machine learning related to the physiology and related to the whole project. Our group doing the electron microscopy on the same tissue that I'll show you the physiology that done at Baylor. And then finally, Sebastian Sung's group that I mentioned doing the segmentation, and all of us doing the data analysis at the end. Here we go.
Well, I'm not sure. The worm has been completely
Yes.
done, and it's a much simpler project. But it must be informative in some way to give you a guide of what's happening, or is it completely a different animal? I mean,
Yeah. So I've given an absolutely, except for one exception, with one exception, corticocentric history of connectomics. The field of connectomics really began in 71, 73, around then with the publication in the early 80s. Sydney, sadly the recently late Sydney Brenner's group, who did serial section microscopy of the worm. What was the title of the paper? The Mind of the Worm or something. A couple hundred pages in Proceedings of Royal Society. A couple hundred pages for a couple hundred neurons. 302 neurons, some number of connections amongst them. Technically, it has been inspirational. In terms of network structure, the different species, worm to cortex, there's not much for me that inspires my thinking of cortical networks in that. I mean, it's extraordinary work, and the worm is an extraordinary model for, it's qualitatively different. And the simplest way to say it is my favorite reason for studying the cortex. I think, many people think, that the cortex is nature's greatest achievement because in neuroscience, because it's a general purpose learning machine. It's modular, it gets inputs, it does stuff, it sends outputs, and it learns extraordinarily. So the same bit of cortex can do different, radically different things. Versus an animal with 302 neurons, which is nature's most perfectly evolutionarily tuned box of hacks to survive. So that's sorry. So here's the physiology from that experiment. But in all these, yeah, please. Many of the same neurotransmitters, but that's where it stops. Action potentials, distinct axons and dendrites. Almost anything you can think of is different. Again, sorry. The connections are different. There are a lot of differences in detail in anatomy and physiology. So here is 70,000 neurons flashing away, not all simultaneously, but collected data over the course of a while. this is the denominator of this big experiment. The physiological activity of a bunch of neurons in 4 visual cortical areas in the mouse while it's seeing stuff, natural movies and other stimuli.
In this image, how do you figure out which neurons are connected?
Yeah, it sounds hard to have, let's call it a constellation for a good reason, a constellation of neurons in 3 dimensions, and matching that with the data set that looks totally, different modality, et cetera. The hard part is making the markers and keeping your wits about you and finding the right bit of brain. But especially using blood vessels as fiducials. Blood vessels are big and then small, then smaller and smallest. Once you can get a rough lineup, and once you're there, you're there. Think of one constellation with 2 telescopes. Yeah, let's match it. Oh, we matched it. You're not going to get it wrong. You don't get it wrong. There's some small number of things with very close together cells here, which you say I don't know, but by and large, it's compared to the other hard problems, that's an easy hard problem.
Is there a North Star that you start with?
The North Star is the biggest vessel on the surface of the brain. You start there, and then you really just morph around in 3D with nice tools. The blood vessels first, and then once you have the blood vessels, you'll see the cells nearby. Then you do some fiducial, randomly chosen North Stars as fiducials, and then it's. Yeah, exactly. Blood vessels are important. So for this project, 3 data sets, one small one, 100 micron scale, one bigger one, 1 millimeter by 1 millimeter, but only 50 microns thick, just to flex our muscles and get used to doing big things. And then the big one, the data set of a cubic millimeter. Very quickly, this is mostly taken from the paper that I assigned of techniques in the field, electron microscopy and then segmentation, calcium imaging first, functional imaging first. To do electron microscopy, you sacrifice the animal, you fix the tissue with chemicals, you embed it in plastic, and at that point it's stained with heavy metals. Heavy metals are the contrast agents that they deflect the nuclei, deflect or perturb electrons, 100 kilovolt accelerated electrons that are focused to make images. What do you make images of? Not of this big block, but of 40 nanometer thick, ultra thin sections, literally called, technical term, ultra thin sections. And essentially it's a diamond knife, the sharpest knife you can imagine, cutting off 40 nanometer, huge, football field size, saran wrap, relative thickness sections that are floated on water, picked up on a substrate, all done in an automated fashion. So we have, it's on tape, a technique Jeff Lichtman invented, and Ken Hayworth invented the first version. Wei-Chung Lee, who was in my group and now has his own group at Harvard, adapted the technique for the variant we use. Essentially, we have 25,000, really 27,000 serial big sections that are on tape that essentially we put inside a microscope and go frame by frame by frame like a movie, looking at these sections and taking a crazy number of pictures. Those are the people who did the various things. And then finally analysis I'll have time to talk about analysis. So phase 1 again is fairly small, 100 microns. Why do I show it? Just to something I didn't really need to show given my powers of 10. exercise at the beginning. 100 microns, if you make it 10 times bigger, it's 1,000 times bigger. And that data set is the one that we collected and preliminarily analyzed by Sebastian Sung. This is an electron microscope. I think I'm not going to go through the details other to say then the sample goes up here. There are reels of each reel has 5,000 sections, and over the course of 6 months we collected 150 million pictures that were stitched together in 3 dimensions. To create this data set, let's skip the details. Skip the details. 5 microscopes. Again, let's skip the details. It's just a lot of data collection. Now that it's in the past, we don't need to talk about it. It's stipulated. One can collect data like this. Let's discuss what you do with the data. Here's the data set at a very, very zoomed out, a little bit more than a millimeter, a little bit less than a millimeter. The only thing you see are blood vessels, the white things. There's a nice big North Star Road, big vessel, and a bunch of little vessels, and those little dots are nuclei. This is what you get out of it after segmentation. This gives you a sort of the 3-dimensionality of it. We're zooming in XY, but we're also stepping through in Z. And you can see the blood vessels coursing along in 3 dimensions. You can see lines going up and down. Sorry about the moire. Each one of those little squares is a 20 megapixel image. Now you can begin to see things coursing up and down. Those are the biggest dendrites, the biggest receiving wires. Now we're going in to see some of the sad artifacts, but our friends in machine learning can get rid of most of those. Now you can really begin to see that's a big axon going in 3 dimensions, et cetera, et cetera. You'll see the data later on with the segmentation. That'll give you a much better sense of what's going on. So we're finally almost at the place we wanted to be 15 years ago. We have very large data sets. We have it segmented. Again, the big data set is not quite usably segmented for most purposes. What can one do? The reason I got into this business, and many of us got into this business, is, quote, functional connectomics, comparing function and wiring. We'll get there, but why do I say that? Why are we doing that? First and foremost, one can also look at network structure. I had sort of a short talk, and I was going to give a coda about network structure, looking at whether networks might be random, small world networks, or whether they might have some low-dimensional structure. Maybe in our open discussion we can talk about the kind of analyses one might be able to do with big networks. Keyword, low-dimensional structures, manifold embedding. I think that if in a network that encodes things like visual space, orientation, direction, or other more derived properties, one can read the connectome with the tools of manifold embedding and extract out the structure of the computation. That was just word salad, keywords, but we can talk about it later because I won't get to that part of the talk. What I will talk about now is cell types. Now that we have this extreme anatomy, we should have had this in our minds at first. The obvious first thing that we can do is really tackle the problem of cell types and their connections. Remember I said molecular determinants of connections, distinct cell types in the cortex. There are 100 cell types in the cortex in any given cortical network. What do they look like and how do they connect? That's what I'll be talking about for the science part of this. I've only had an hour of technical introduction, but I hope it's been an introduction to the field. Even with our small segmentation, we've been able to do a cell types analysis of a small network that's allowed us to ask and answer questions that would have been extremely hard, if not impossible, with previous techniques. And the real hero here is abstractly the field of machine learning and convolutional networks in particular. In the past, as you all know, in the past 5 years, 3 years, 2 years, the advances in machine learning have been revolutionary. And probably the best, but there's so many great things, probably the best has been machine learning for image segmentation, for image interpretation, image processing. And that's sort of recursively, reflexively, what's pushed this field forward so much. We're funded because of trying to find inspirations for machine learning, but the field has been transformed by machine learning very recently, and really the pioneer in this has been Sebastian Sung, using machine learning for segmentation, and very luckily we were paired with him in this project. And now we have, in this small data set, several hundred neurons just beautifully segmented in all of their detail, and with a little bit of proofreading, the connections that each one of these neurons makes. This is a little bit dark. I'll get to a bunch more later on. Okay, here is where I hope you can appreciate a little bit better the electron microscopy, when it's the coloring book version or the colorized version of the movies, the black and white movies that you just couldn't interpret. But with machine learning, each individual element, nuclei, cell bodies, big dendrites, small axons, etc., are segmented from, each voxel is assigned to a neuron, to a connected region. And this is a lovely tool. You're going to see a movie using a lovely tool that people at Google developed. Google has a very big team working exclusively on microscale connectomics doing this. And one nice thing they've done is to make a tool that all of us use now. So again, this is going through in 3 dimensions. This is the data on the left. On the right, you'll see neurons. This tool is really fun that you can just click on any one of these things and almost immediately get its 3-dimensional structure. It's a small data set, so we don't see an entire neuron, but it's extraordinary. I like to call it OneClick Golgi. The Golgi stain is where individual 1 in 1,000, 1 in 1,000,000 neurons was labeled, so people in 1890 could see individual neurons. Now we have OneClick Golgi. We can click anything and see the neuron at essentially 4 nanometer resolution in X and Y, and 40 nanometer resolution in Z. Again, it's kind of a small data set, but you can just see the detail with which these things are reconstructed. So given that we have these gifts from 21st century computer science, what do? What we have started with, starting with the team at the Allen Institute and collaborating with Google's team on all of this analysis, is get individual neurons, have all of their synapses automatically labeled, and just look and classify. This is probably the prettiest neuron. It's an inhibitory neuron with 4,700 synapses onto it. 1,700 output synapses. It's not complete. It's bigger than that, but it's pretty complete. Here are 5 neurons, including the original, of different inhibitory classes. There's a real zoo of inhibitory neurons. Excitatory neurons look the same, although there are many subtypes, both according to layers in the cortex and molecular things, molecular labels. I'm very quickly going to go through the most neurobiological story, which is just to give a sense of what one can do if you're an anatomist, a nerdy neurobiologist, and say, I want to understand one class of neurons and the connections it makes. One of the most idiosyncratic and just interesting neurons is known as a chandelier cell, or as my entire team, mostly from Europe, chandelier cell. One second. And these are inhibitory neurons that do one thing. They send out axons that terminate not on a dendrite, but they terminate on the very beginning of an axon of an excitatory cell. And they typically terminate in you'll see, I think, in a series of synapses, all up in a row. Looks like a candle. So the whole thing looks like a chandelier with these little candles pointing up. Question? A limit of, yeah, do we understand where the limit comes from? Not really, but they all, each cell class is different. But take a pyramidal cell, an excitatory cell. Take an excitatory cell. They send out between 5 and maybe 7,000 output synapses, receive roughly the same number of input synapses. And those are pretty solid. You're not going to have a pyramidal cell that sends out 100 big synapses ever. But the structure of the network is important. And there's no rule that says if you have 2 neurons, that neuron A doesn't give 30 synapses onto neuron B, where the average coverage is only 1 in 10 targets. So it's decidedly not Poisson or random. There's concerted connectivity, if that helps you. So the rough numbers of in and out are the same, but the numerosity between any pair is boundless almost.
This limit or this structure of the network, you think is coming from a particular reason, or if it's a network connectomic like a circuit like?
Well, not quite chicken and egg, but it probably doesn't come from molecular reasons. It probably comes from physiological reasons. Why they're mostly zeros. If there's 1, there tend to be 5. So the statistics come from obscure sources, but the current hypothesis is that that at that scale, what is the structure? So let's stand back a bit, and this is actually, I should have done this early on. Let's just consider the excitatory network, okay? What are the, I've been dealing all in powers of 10 rough numbers. Each neuron, for physicists, each neuron gives out 10,000 connections, receives 10,000 connections. The numbers work out that the probability of any neighboring neurons of being connected is roughly 1 in 10. Okay, so it's a densely interconnected network, but it's a sparse, by pairwise it's sparse. And there's a lot of theory out there. This is sort of interesting that given that numerology, might this be optimal for X, Y, or Z, optimal for learning, optimal for synchrony codes, optimal for anything you could imagine. And it has to be optimal for something. Evolution made it so. And it's a really interesting question. Now that we can finally see not just the stupid numerology of 10,000 in, 10,000 out, 10% interconnected, but the real statistics, we can pose a question. How many bits per synapse? What that might mean about learning rules and coding. So. Yeah. So do things change? Absolutely. And again, I gave one. This is not. It's beginning to sound pejorative, but isn't. One for physicists' view that connections start as dense and promiscuous and get pruned to be all sorts of things, but more specific along different dimensions. So that's over development. The development has multiple stages, sort of early development, critical period when a lot of things are laid down more solid, and then adulthood. Clearly, we learn. I hope we learn. I hope I've changed 7+ synapses this morning, and I hope most of those synapses have been in the cortex, so you can remember, not in your amygdala, to say, I'm mad because I didn't get it. So a lot of synapses change in the cortex, have to be, and that's a huge thing to study. Dogma, not true anymore, is that the synaptic structure of certain cortical areas, like primary visual cortex, is tuned up during the critical period that ends early in development, early in postnatal development after animals have seen, and then remains relatively solid forever. Not remotely true anymore, but that's one caricature. There has to be, again, I've said it, a great deal of cortical plasticity in us, whatever our age is, and it's a field of study. So, just by developmental connectomics, neuroplasticity connectomics, these I hope are fields that'll happen. I'm glad I left a lot of room. I won't get to the second talk, but so anyway, chandelier cell. This is, even for a small data set, we can actually study a decently complete chandelier cell connectome, because these cells only target the beginning of the axons of excitatory cells. So just as a first study from our small data set, we took 100 some odd pyramidal cells near the top of this sample, all of which have these initial axons, and said, let's look at every single synapse onto all of these axons. And what we found was, classically, thank goodness, we found that there are chandelier cells that make a classic candle with multiple synapses. This blue thing, I think I'll have examples later on. This blue thing is one chandelier cell that makes multiple synapses onto this specialized portion of the postsynaptic cell. This is I have to show one summary plot. This is one summary plot of length versus count. 0 is at the cell body, the axon going down 50 microns. The red histogram is chandelier cells. These are over 100 cells or some number of cells. This is a histogram of all of the chandelier cell inputs to axons for this set, and there are other types of inhibitory inputs in gray. So as the literature hinted, as literature said, but really now we finally have a beautiful histogram, that there is a sweet spot in this specialized portion of axon that collects synapses. I'll skip this. And what was really fun in this, we decided this is a classic cell. There are a lot of papers, a lot of questions about this kind of cell. Let's study it. And we were very pleased to find something that's hinted at in the literature, but really not shown like this, that these axons in magenta-ish, whatever that color is, these axons actually look extremely plastic, extremely changeable. There's a thing called a growth cone, which is something that forms on axons that are trying to find targets. Axons are these wires, but when they're looking for targets, they put out fingers, essentially. If you see a movie, they look around for a target. You don't see growth cones almost at all in mature cortex, but here, when you look at this population, there are actually a lot of growth cones. So clear evidence of a lot of plasticity in this connection. And also fun things like, in fact, the most common connection is only one connection, not a whole candle. Sometimes there are several, sometimes there are a whole bunch. Often you have these anthropomorphized searching growth cones, trying to find new connections. So it's the kind of thing that is either depressing or exciting. I choose exciting. That the connectome is huge. It's not a connectome. It's not a graph. It's really all of anatomy, plus the network structure. And collecting it is not getting one datum, but it's creating a data set that one hopes almost anyone in cortical neurobiology or neurobiology of an entire species will look at. So, especially being at the Allen Institute, where the Allen Institute was founded 15 years ago by Paul Allen and his sister Jody Allen, essentially to do big projects in biology that academic labs couldn't or wouldn't do, but to provide resources, web resources that could be used by neurobiologists. Now there's also an immunology and a cell biology, cell science institute, but there's the original Brain Institute. And what it's done from the beginning is to put out resources, mostly atlases of gene expression in the mouse brain, things that neurobiologists can go to and do science. This is something that we hope, with the tools that we're developing, that people can go to, ranging from ultra specialists who are studying axon initial segments, et cetera, to physicists and network scientists who want to understand the structure, topology, manifold embedding structure of networks. So really it's a resource that if the tools are good enough, that almost anyone can use. And what I'll finish with is sort of the tools that we've developed. Yeah.
One of the limitations, obviously, of deep learning and neural network is you're going to get associations. You're going to get correlations. One thing turns on, another thing turns on, one thing turns on, another thing turns off. But not causations.
Yeah.
Is there some way, and I imagine those associations that are functional will have some reflection in anatomical descriptions that you have, and some that will not. Actually.
Yeah.
And so you'll come to a point, I can imagine you coming to a point where one layer deeper to understand causation and interactions and learning how it all adds up is the next challenge.
Yeah, I mean, there are a lot of next challenges, and there's a lot in the question. Let me, you know, let me finish.
Yeah.
But let me say one thing, because it's easy to listen to a talk like this. It's easy to hear the word connectomics, omics, and say we're searching for a GUT, you know, a grand unified theory of how anatomy causes physiology, how learning rules cause anatomy. And yeah, I hope so. But that's not what we do from day to day. And you know, in all of biology, we see correlations and almost, you know, never see a causation. But you know, with such rich phenomenology, you can get closer to a lot of difficult things like those. But this is not an exercise in creating a model of the cortex that predicts everything. I guess that's the important thing. I mean, it's obvious, it wasn't even in your question, but it's good to say. At the Allen Institute especially, we're interested in cell types. I really don't have time to go into that, but there's a whole program called cell types that this is a part of at the Institute, at least, where we study and sort of inside baseball. Inside joke almost is that we're doing cell cards like baseball cards, but I think this may become part of the product that if you want to study cell types, you have a card that will, you know, give a picture of it, like the front of a baseball card, and the back might give you various types of summaries, one card for each player. What are we doing for analysis in the last 10 minutes? Using the segmentation and starting first with just well-trained biologists' eyes and brains, you know, making inferences and you know, deciding what to study, but really then going quickly to machine learning approaches to classify, find patterns, etc. And the easiest thing that one can do is start at the nucleus and just look at nuclear morphology and ask, can you do machine learning on nuclear morphology, or just statistics regression on nuclear morphology and classify different cells? One thing we do is that's what we can do without any proofreading, without any human work on the segmentation. Then we can do a bit of proofreading because it doesn't start out as perfect, these segmentations, and you know, finish the axon and finish the dendrite morphology, finish the axon, and then finally do science from functional connectomics through network science, and just the flavor of you know, the easiest thing one can do is, again, taking nuclear morphology and just plotting things such as the volume of the nucleus versus the surface area, which, you know, the relationship between the two gives, you know, the deviation from how spherical it is, and see that there are distinct clusters, and those distinct clusters very well predict which cell, which type of cell it is. You know, there are excitatory cells. There are these amazing astrocyte cells that fill in all of the interstices that look like, you know, fractal sponges, different types of glia, and you can classify them with the simplest morphological criteria. I'll skip that, do it again. But really going into just the beginning of the rules of cell type connectivity and the structure of the network. I'm going to show another movie of one fairly typical, actually, excitatory cell and its inputs. Is this a movie? Or is the next one? That's a movie. So, it's three-dimensional. The blue things are in the inhibitory synapses. Let's turn off all the excitatory. And they're the largest class of inhibitory cells are basket cells. Those are all the basket cell inputs. The chandelier cell inputs that you know and love, I hope, are right there in one part. There's another class of inhibitory cells, somatostatin cells, that are sparser inputs and what happened? Anyway, that target different parts of the cell. So very quickly, an expert can go in and ask very specific questions that just really would be almost intractable without this kind of data. I'm going to skip this other. No, I won't skip it very quickly. In an almost automated fashion, not quite yet, one can classify the different types of inhibitory axons and then ask what targets, what is the target of these different type of inhibitory axons. Again, a question that is very hard to look at with other techniques. And this is a very, as you, I hope, appreciate, it's not a walk in the park collecting these data. But once you have it, you can ask questions just one after another about, in this case, cell type specific connections. Each column is a different class of presynaptic neuron, inhibitory cell. Each color is a different type of target. And I won't even go through what the different types of targets are. But as you can see, there's a great diversity of these barcodes of different targets. So, a figure like this, which took, a month or two of work and now is almost a query, would have been the summary of a large number of studies previously. And then finally, I'm just going to end with, it's all in Python. Everything that you saw of the neuroglancer tool of going through the data, clicking, etc., and that you haven't seen of annotating synapses, excitatory, inhibitory, location, etc., all that has a back end, and you can do simple Python, SQL queries that have semantic labels of types, but also XYZ labels of where they live. That is linked into really I think the most important thing is the three-dimensional structure of the neurons that live as meshes, as little tessellations of the surface of these three-dimensional structure. So you have just much more than a connectome. You have just every bit of information that you'd want. If you're interested in the functional geometry of closely packed networks, you got it. If you want to just look at the network with various levels of abstraction, excitatory, inhibitory, you got it. If you care about 30 classes of inhibitory cells, you got it. You got it in quotation marks. Don't go today looking for this. It's not ready for prime time. The cubic millimeter is not. It's segmented, but it's not there yet. But we're a long way from 2004 when we just had these wonderful movies. I will leave you with the grand challenge of going up merely another factor of 10 to do the whole mouse brain. But again, a factor of 10 is a factor of 1,000. It's really a factor of 500 or 400 times bigger than the data we've already collected. I think many of us think I'm going to a hush-hush meeting. I won't say it because I'm being taped. I'll tell you at lunch. We're talking about we're talking about, we being the community, talking about doing the whole mouse brain. And the NIH has come out in its most recent Brain 2.0 position paper saying this is a grand challenge that it's time to do, and they're planning to fund it or planning to consider it very seriously, this huge project. This is the Allen Institute. The current picture is even more people. This is our nice building in Seattle. And always thanks to Paul Allen and Paul and Jody Allen, our founders, for vision, encouragement, and support, and so much more. Coda, you can look at it, it's pretty. Questions?